Okay, everybody, I thought that it would be easier if I just walked you through this PowerPoint than just having you click through, make it a little bit more interesting. In this particular presentation, the focus is understanding what we use nanotechnology and nano devices for, and then go through a couple different examples of things that are in the process of being made now. To give you some perspective, a nanometer is crazy small. Nano, by definition, means times 10 to the negative 9. When we're using pipettes, we are on microliter scale. That's -6. So you can see in this particular scale of things, a water molecule is times 10 to the negative 1. And then when we go even smaller than that, these are getting smaller and smaller and smaller. As you go left, it gives you a function in nanometers. So glucose molecule is about 1 nanometer in width. An antibody is about 10 nanometers. So when you're talking about viruses and bacteria, they're about 100 nanometers versus 1,000 nanometers, and so on. When you're talking about specific devices that we're going to be talking about later, it's really on the order of about 10 to 100 nanometers in terms of diameter. And when we talk about the size, a white blood cell is about 10 to the 4th nanometers in size. So thinking about how large that is, you can fit a lot of small nano devices if they're on the order of 10 to 100 nanometers in size into one particular cell. So the idea is we make these things small enough so that they can go inside cells and maybe target specific elements within the cell and have an impact. And since we're learning about cancer, typical applications for this might be a targeted drug therapy. So making sure that it gets to cancer cells specifically and that it can actually get into the cancer cells and either have some kind of drug response, or maybe it's for visualization purposes, tracking cancer cells and where they're going and how many there are, things like that. It's important to realize that when you get to that small of a scale, things start to act differently. They have kind of different properties than they do when we get to the macroscopic scale of things that we can see. Because they're so small, they have a ginormous surface area relative to their volume because they're so small. So things like friction are more important than they are in macroscopic type things. So things behave differently, they bend differently, they act differently when you get down to that small scale. And so you have to kind of accommodate for that when you're designing nanotechnology. Another thing to think about in terms of nano devices, the body can probably clear them rapidly. Because they are so small, your body can kind of get rid of them quickly. And if the goal is to detect something or image something, then that might be a problem. Another perhaps unintended consequence might be that they clog up inside of cells of vital organs. We don't really have enough knowledge of using these things in people to have an understanding of how they would work. So it's possible that they might begin to accumulate in certain organs, and maybe it will cause a toxic problem. So the idea is to create something that will go into cells and last long enough before getting cleared away, but then also not stick around and clog things up and cause problems. As I mentioned, detection is one possible thing that this could be used for. So depending on what you want it to target or track, it is really able to show you very fine-tuned cells. So maybe it's showing you the very, very earliest possible stages of a cancer development. Whereas on typical imaging that we have now, you'd have to have a mass of some kind to be able to see it, or wait until someone is actually experiencing symptoms. Here's just an illustration of just how it really can improve sensitivity, showing how they can determine depending on what cells they're targeting, maybe precancerous cells versus normal cells, or start to accumulate in particular cells so that you could visualize them. Traditional tests really require you to see advanced stages of cancer or cells that have been altered, and you actually have to do a biopsy. You have to take those cells out of the patient and preserve them on a slide and look at them. And so you can't, use those again. Whereas in nanotechnology, if you can track cells in the living patient, you don't need to remove any, and you can actually track them over time. You can do additional tests, things like that. As I mentioned, they can make cancer tests faster, more efficient. You can also run many, many tests simultaneously. Which leads me to the specific different devices and their applications. You will have to know, generally speaking, the different categories and what they are for. So a cantilever basically is a tiny lever. It looks kind of like an E with an extra prong hanging out of it. And those little extensions or prongs that are hanging off the major side here can bend. So the idea here is that you might attach specific antibodies or specific proteins, something that would react or try to detect a product of a cancer cell. So whether it's looking for altered DNA, or maybe it's an antibody to a specific protein that the cancer cells only would produce, then really what happens is the cancer cell would produce the protein or produce whatever agent is going to react and attach itself to the appropriate lever arm. We learned previously that antibodies are highly specific, so you can even design a cantilever to have different levers have different antibodies. So you could be testing maybe for 4 different things in one cantilever. And then whatever binds, that extra weight causes the cantilever or the lever arm to bend, and then you can actually see that response. So in essence, it's kind of like a mini ELISA test for cancer. If you think all the way back to Unit 1, we tried to detect Sue's meningitis antigens using specific antibodies. And pregnancy tests work in a similar way. This is a nano-size scale that might be able to work. When the cantilever has the molecule bind to it and the cantilever bends, it might flash a light or trigger visualization in some way so that the scientist could actually see it. And so by looking at which levers bend and to the degree that they bend, you can gather information about cancer cells or whatever cells you're trying to detect. And that's basically summing up what I mentioned. It's able to detect very low concentration, so it could help you detect cancer early. The next one is called a nanopore. These are tiny holes, and it's basically a sheet that has a whole bunch of holes in it, like Swiss cheese, and it's just small enough so that DNA can pass through one strand at a time. Why would we ever possibly want to do this? Well, it helps to improve the idea of DNA sequencing. If it can only feed through one strand at a time, it makes it a little bit easier to read the letters or the nucleotide code as it passes through. So it can detect the A's, T's, C's, and G's. And each one of those different nucleotides has slightly different chemical properties, so you can actually detect which one is which. It can also detect potential errors. If it's reading that code and it has a comparison code, it'll be able to show you changes and maybe then link errors to some kind of cancer mutation. So here's roughly what that looks like. You can see that there's a single-stranded DNA molecule going through a nanopore, and it's feeding through at a typical Speed, it's able to detect the chemical properties of the different bases, and then could read like an A and then a T, and then you could get a computer readout of what those would be. As I mentioned, each base has slightly different properties, so you can actually see changes based on those chemical property detecting, I guess, sensors of the nanopore. Okay, the next is a nanotube, which, as you can imagine, is a tube. It's super small. It is half the diameter of a molecule of DNA. So think about it's as wide as about half of the ladder. And it's made mostly of carbon. And it can detect the presence of altered genes and pinpoint specific locations. It also has a huge structural component. You can use them to build different things, kind of like a cytoskeleton. But here's roughly how it can mark a mutation. Scientists might attach a specific huge molecule, like a tag, to a known mutation. And then if that DNA gets kind of stuck in the nanotube, then you can detect if a person has that mutation or not. The tag would only stick if the person has the mutation, and then when you feed it through the nanotube, then you can actually see if that person has the mutation or not. We've seen tracking molecules before. When we talk about radioactive tracers and imaging, we are able to find tumors, look at blood circulation, and do a whole bunch of other things. But when we go on the nanoscale, it's much more refined, much more fine-tuned. I don't know why that slide was just there, but it's relevant later, because then it's kind of circling back to nanotubes for whatever reason. The imaging piece goes along with quantum dots. In this particular case, a quantum dot is a super tiny crystal that glows under UV. So if you think about all the different UV glowfish or UV light bulbs you can get, things can be illuminated in many different colors. So quantum dots exist in a number of different colors. You could use all of one, you could use many, but the color with which it reflects is dependent upon the size of the crystal. And then the idea is that normally they don't have a color. They would go and infect particular type of cells. Let's say we're targeting them to cancer cells in some way. And then you turn on a UV light for visualization, and then all of the cancer cells might light up a certain color. In terms of targeting them to cancer cells, you can have them be designed to bind to a specific DNA sequence. So if you wanted to look at DNA or you're trying to target cancer cells, that's kind of how you would go about doing it. And then once it's bound to that DNA, it's kind of in the cell. And when you flash the light on, then it's going to glow a particular color. So you could see where the cancer cells are. Has it moved to other locations? How big is the tumor per se? Those kinds of things. So the advantage is that you could have lots of different unique labels. You could target different size quantum dots to different locations. So you could actually have multiple colors and track movement in a live person over time, which is pretty amazing. And you wouldn't have to actually remove any cells from the person, so that eliminates the need for a biopsy. Another cool thing is called a nanoshell. These are like tiny little heat rocks, so to speak. So they're little beads and they're covered with gold. Don't worry, they're so small that the gold is pretty negligible. But then you have them target specific cells. So eventually, in a perfect world, they would make their way into cancer cells. And then the idea is when you shine an infrared light on them, kind of like a microwave, then it heats the gold up and creates this intense heat in a very localized area. So it basically nukes the cancer cells from the inside out. Those nanoshells heat themselves up and then kill the cell that they're in. It's a much more effective treatment because you're really killing the cell within instead of trying to take out a whole area of cells or your whole body with chemotherapy. Here's kind of a visual of what that might look like. You have the nanoshells, they attach themselves to cancer cells, you heat them up, and then the intact healthy cells around remain happy and healthy. So we know how already to link nanoshells to antibodies that recognize cancer cells. So if we can do that, similar to ELISA way back in Unit 1 when we had a secondary antibody with an enzyme. In this case, it would be an antibody with a nanoshell attached to it. And if it can attach to a cancer cell, then we're doing great. And then in lab culture, it's working. So we just have to get to a point of actually being able to maybe use this in people. The last one is called a dendrimer, and this kind of looks like a dendrite of a neuron, which is how it really gets its name. It's a super branched-like structure. It's a man-made molecule that's about the size of a protein, and the idea here is to just create an insane amount of surface area compared to volume, with the goal of drug delivery. So if you can attach drug molecules to each one of those branches, then that super high surface area can help in the drug delivery aspect of cancer treatment. And here's just kind of a visual. It doesn't really do anything per se. But it can act as a therapeutic agent, meaning drug delivery. It can act as a reporter if you have some kind of fluorescence attached to it. It can monitor cell death if it's programmed to attach or respond to specific proteins that are involved in apoptosis. It can detect cancer. So it has a lot of applications in terms of delivery and detection. And here's kind of what I just went over. So overall, the goal is to try to minimize cell death, to really try to target and destroy the cancer cells only, leaving your healthy cells intact. And much of our current treatment can't quite ensure that all healthy cells are remaining alive. And also to really advance the idea of imaging. So as I said, we kind of have crude imaging. We can track things over time, but usually we can't visualize it until it's already progressed. Nanotechnology offers an opportunity to detect things earlier when things are smaller and more localized. We could detect precancerous cells before they even become cancerous. We can do drug delivery at very cell-specific spots. Those are some of the hopes that as we continue to develop this technology that we could use in humans. And this is the last slide, but also think about, as I mentioned previously, the drawbacks that might be involved with nanotechnology. It's very new. We do not know what it would do in humans, whether it will be cleared away too quickly to be effective, what the cost of this kind of treatment might be, what are the long-term consequences on the human body? Will these things accumulate? Can they cause their own problems over time? So, though this is a super promising, plausible way to intervene in cancer moving forward, there are some ethical considerations that you have to take into account as well.